Iamana, ingareo, rangitirama, tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Andres Matisoo toku mātua no Estonia ia. Ko Julia Moores toku whaia no United States ia. Ko Brent Smith takutane no Tamaki Makaurau ia. Ko Lisa Matisoo-Smith takuingoa no Hawaii ahau. Ko Otepoti taku kainga inaiane. Nō reira, tēnā koutou, tēnā tātou katoa. It's a privilege, an honor to be here today, and to be able to tell the story of the work that I've been privileged to do over the last 30 years since I arrived here. And we've heard many stories from the anthropologists, from the archaeologists, from the oral traditions, about the languages that link us all together. And now I'm adding the DNA story, which is another thread that we use to really tell the rich stories that are our human stories and link us. We all have distinct DNA, but we're also all linked together through that DNA. It's our shared history, and also it links us to all living things, our DNA ancestry. And so we have that responsibility to look after each other and to look after all living things. And I think that's, what's happening. These conversations that we're having here today are part of that. So here we go. I'll try to stay to time. We're terrible academics, and the more senior the academics, we seem to go on forever. But I'm going to tell the story about the DNA kind of mapping. It provides a map for us, a signpost of the points of the journey that we've all been on. But it also is impacted by, it's not just a straight up map that anybody can read. It's always influenced by the environment and all of the various things that we encounter along the way. And so that's the story that I'm going to start telling today. We've already heard a bit about our shared origins, our African origins. And it was actually a great New Zealand scientist, New Zealand-born scientist. It was at the University of California at Berkeley, Alan Wilson, who recognized the power and the knowledge that's carried in our mitochondrial DNA. That's DNA that we inherit from our mothers. So as a woman researcher, I quite like the fact that I'm studying the maternal histories of human migrations. As we know, men can spread their seed around, their DNA around. Women, for a mitochondrial DNA lineage to be present in a place, it means that a woman gave birth to a female child there, and that lineage is maintained in that location. So it's a connection to the land through women. So here we go. This is the story of Africa to Aotearoa. We all started in Africa, and as we've heard today, Aotearoa was the last landmass that humans settled. And I think one of the greatest stories of human history. So we've already heard about our Austronesian linguistic origins, linking back to Taiwan, the homeland, the Hawaii of the Austronesian languages. And you can see the Pacific languages represent the oceanic subgroup of the Austronesian languages. And we've heard about the Austronesian expansion, originally starting on mainland Asia, then out into Taiwan, as a result of the development of rice agriculture and population increases, we think, on the mainland then, about 6,000 years BP, years before present. And they passed through island Southeast Asia. And traditionally, we have the story of the Austronesian expansion arriving in the Pacific and associated with the Lapita cultural complex. So we have the language now and the culture, the material culture that we see in the archaeology. And the Lapita culture first appears in the Bismarck Archipelago in what is now Papua New Guinea and spreads across into the region we refer to as remote Oceania, where they are the first people to arrive. And they reach the edge of the Polynesian Triangle Samoa and Tonga. So here is again that Lapita dispersal. The earliest dates we have for the appearance of the Lapita cultural complex are in the Bismarck Archipelago and date to about 3,350 years ago, years before present. Almost within a few hundred years, very, very quickly, we have people arriving in the Reef Santa Cruz in the Southeast Solomons. The first islands that they encounter where there have not been people living for tens of thousands of years. We believe that they are the first people to arrive in the Reef Santa Cruz, Vanuatu, and New Caledonia about 3,000 years ago, 3,100 years ago maybe. And by 2,850 years ago, they're on the edge of the Polynesian Triangle. They stop there. Lapita expansion stops there. In Tonga, lots of evidence of Lapita occupation settlements in Tonga, and less so, but some archaeological evidence, Lapita pottery, and people arriving in Samoa very much the same time as Tonga. And this is the Lapita pottery that is the signpost for archaeologists, I suppose, of a Lapita site. But of course, Lapita sites, there are a number of artifacts and settlement patterns and other characteristics as well. We have the classic Lapita face, but what we can't tell, while we see the face of Lapita, we don't necessarily know what the faces of the Lapita people looked like. Your DNA does not determine your pottery or your cultural characteristics or the language that you speak. So we have to remember that these are all different threads of a story that might not tell us exactly the same thing, but come together to form the complex story that is our story. So then we have the last burst out, the last expansion from the Polynesian homeland that's always been described as Samoa, Tonga, those Lapita-derived populations. Then after 2,000 years or so, and the dates are regularly being disputed and discussed, but for a while now we've pretty much accepted that about 1,200 years ago people moved out of Samoa and Tonga or people arrived in the Hawaii zone for East Polynesia, the Cook Islands, the Society Islands, the Marquesas, and from then up to Hawaii, maybe 1,000 years ago, out to Rapa Nui, Easter Island, about 750, 800 BP, and down here to Aotearoa, about 750 BP. So interestingly, why did Lapita stop has always been an interesting question. And then why did we have this burst out again? And there's been some interesting debates. There's no pottery when people come out into central East Polynesia. They're not bringing pottery with them from that Lapita pottery society, post-Lapita. So it's been an interesting question. Now, we've also heard a bit already about the transported landscapes of the Lapita people and Polynesians carrying their important plants and animals and ideas and things with them in their waka as they dispersed across the ocean. And so I was very lucky when I arrived here in Aotearoa and started as a young anthropologist. I was trying to find some interesting questions, and one of the first people that I really met are here in the room today. Richard Walter, who was in the anthropology department. They dragged him back from the Cook Islands, and he was finishing his PhD when I arrived. And Meta Roberts was working at the university, and she and I were teaching a course together. And Mere was doing her PhD on the Kiore, and she introduced me not only to the Kiore, but I'm very, very lucky. She introduced me to Hori Parata from Ngāti Wai and Kaitiaki of the Kiore. And this was at a time in about 1990, 1991, that the Y262 claim was being submitted, and both Mere and Hori very kindly shared much of their knowledge and information and taught me. So I thank you both, and I'm very humbled that you're sitting here while I'm up here talking right now. So I was introduced to the Kiore, Ratus excellens, and it's just a rat, and it's so much more than just a rat. And also, sorry, I would like to acknowledge two other people who are no longer with us, but I'm sure are looking down on this and smiling. I can hear Roger's laugh right now. Professor Roger Green and Professor Ranginui Walker, who, as it said, we stand on the shoulders of giants, of our ancestors, and these are the people who are why I'm here today. So I acknowledge them. I thank them. There was one other person we were saying we haven't really acknowledged much, and I think we should, and I was very excited to hear him speak, and that was Ben Finney, who in 1987, the first conference that I went to here when I arrived, and it was a student, Ben Finney was speaking and talking about the voyaging and the regeneration of the voyaging tradition and voyaging knowledge. So I'd also like to acknowledge Ben. But back onto the Commensal story. What Mere and Hori were explaining to me about Kiore being brought here, and that it was a taonga. It was brought intentionally, and it was, a very Eurocentric view that rats are stowaways and bad and nasty things, that why would anybody take a rat? And so I learned, this other way of looking and different relationships and ways of interacting. And it made me realize that these are the animals that travel in the waka. If we could figure out where they came from, if we could figure out where the Kiore from here came from, then that's telling us where the waka came from. And it was a way of tracking by looking at the plants and the animals and the insects and all of the things that came in the waka. We have this other way of tracing the movement of people, through the Pacific. And so this commensal approach, commensal means eating at the same table. These are species that are closely related to humans. And there are debates about intentional and unintentional introductions, but different species are being brought, and many of them, including the Kiore, intentionally. The kuri arrived here. The pig and the chicken were two Pacific commensals that we see in archaeological sites across the Pacific. And so I realized that we could use this mitochondrial DNA that Alan Wilson and his team first published in 1987 in terms of human DNA. So this was just at the time that we realized we had this new tool. We could look at mitochondrial DNA. The Kiore also have mitochondrial DNA, and it's passed down through their maternal line to trace the Nohea te Kiore. Where did the Kiore come from? And DNA often just tells us stuff that we already know, and I've had this conversation with many people across the Pacific. We trace the mitochondrial DNA of the Kiore from here in Aotearoa to the Hawaiki zone, to Rarotonga, to Raiatea, to Mere and I were in the back of the Whaaroa Valley, where you have Tapu Tapu Atea at the mouth of the Whaaroa Valley, and catching the rats there. And what we actually found is that the Kiore there have the same DNA, the same mitochondrial lineages as the Kiore we have here in Aotearoa. And people would say, you will find that because the Aotea canoe is actually back in the back of that valley, and it came back and it's buried. It's in the river back there. We'd say, okay, and I got back to the lab, and lo and behold, the DNA was identical. We then decided to take things back further, the origins. Oh, sorry, forgot to say, these interaction spheres. This was a fascinating, again, we've heard this story, so many times today from so many different lines of information about the identification of the Hawaiki zone, about an interaction sphere that stops off in the Kermadex. The Kiore stopped off in the Kermadex as well. Before coming down here, we have evidence of shared lineages that suggest an interaction sphere in the south, this southern interaction sphere, and a northern interaction sphere between Hawaiki zone out to the Marquesas, up to Hawaii, and two-way voyaging between Hawaii and Hawaiki. So we decided to say, okay, let's go back further. Let's look at those Southeast Asian, the Lapita Kiore. We find Kiore in Lapita sites. And so we started looking at both archaeological remains of Kyoto, which we could get the DNA from with the new technology that was developing through the 1990s. And so we could get DNA from the Kyoto from archaeological sites, and we traced them throughout the Pacific. And we found 3 main family lineages, lines, we call them haplogroups in DNA language. We had the group 3 rats that we see out here in East Polynesia, the 3A rats really in West Polynesia. Interestingly, we do have some of those lineages down here. And they were a different family group, but related to the rats here that we see from the Philippines all the way through to the Reef Santa Cruz. And then we had these group 1 Kiore that we found in the Philippines and Borneo and Sulawesi. Now, interestingly, this lineage that we see out here, 3B, we also see back here in Halmahera in the Moluccas, an area that I think is actually a very important place for us to be thinking about. We then realized we could also look at the other animals that people carried with them, and that we started looking at the Pacific chicken. And chicken bones, only with the Kiore, we worked with both modern Kiore that are still living and running around on the islands. And the chickens, we only look at the archaeological remains because European chickens have replaced Pacific chickens in most places. So I had a wonderful PhD student, Alice Story, who was looking at chicken DNA from archaeological remains across the Pacific. Of course, no chicken here in Aotearoa. We had moa. Didn't need those little scrawny things. And while Alice was doing this work and I was talking at conferences about our commensal approach, a colleague of mine from South America, from Chile, Jose Miguel Ramirez, told me that he knew of some chicken bones in an archaeological site at a place called El Arenal on south central Chile, and that's the site there. You can see it's a beautiful, it's a beautiful place to land a waka, nice sand dunes, and there was an archaeological site here that had chicken bones. Now, chicken are not native to the Americas. It was believed that Columbus and the Europeans introduced chickens to the Americas. And Jose Miguel told me this was a pre-Columbian, a pre-European site in Chile, and it had chicken bones in it. And would we look at the DNA of them? So I met him at the airport in Santiago, and I was on my way to Peru. He gave me the chicken bone, which I brought back, and Alice looked at the DNA. And the DNA was identical to the DNA that we were seeing in the chickens from Samoa, the 2,000-year-old chicken bones from Samoa, and the 3,000-year-old chicken bones from Vanuatu, Lepida-associated chicken bones. We found 2 lineages of chickens, and both of them are in Chile. The very early ones that we see in Lepida sites were in the El Arenal site, and some very early historic chicken bones from South America had the second lineage. We had those chicken bones directly radiocarbon dated, and the radiocarbon dates, much to my surprise, came back clearly pre-Columbian, before 1492, when Columbus arrived on the other coast of the Americas. The chicken bones are Pacific chickens that date to pre-Columbian times. So we published this research saying it was the Polynesians who carried the chickens to South America. Again, we knew this because the kumara, which is a South American plant, was here in Polynesia. We have some dated charcoal remains of kumara, but we know that they were present by the time people left Hawaii to come down here. So we had the theories of Thora Heyerdahl with South Americans who sailed out there. No, the Polynesians are the greatest voyagers in the world. Why would they stop voyaging? You know, they didn't have a sign when they got to Rapa Nui said no more islands beyond here. They would have continued until they could sail no more. And that's basically when they hit the land of South America. What's interesting is where they're likely to hit, where we found the chicken bones, is just about here. You can't sail back from that location. You actually have to sail up the coast to the Gulf of Guayaquil in Ecuador. That is where the kumara can be traced linguistically back to kumar in the Quechuan language. They picked up the sweet potato and came back and brought it to Hawaii, and it was distributed throughout the network of interactions. So we published that. We got quite a lot of flack from the Americanists and several others. We've now been replicating that result, and we're just about to hopefully publish another paper on that. People have been doing the DNA of the Kumara as well. The Kumara, we see 3 introductions of Kumara into the Pacific. We see the Kumara from the Gulf of Guayaquil, from Ecuador, coming out, and that is the Polynesian variety of the Kumara. We also see the Central American camote line, and then we see that's coming out of Central America, and then we see also Kumara coming out of the Caribbean, being taken into the Dutch East Indies, and that's the Batata line. So we actually see that it's the Central American Kumara that come down through into New Guinea, as well as the Batata, the Caribbean Kumara. We've also just published, I've got more other colleagues in Chile who I've been working with who've been looking at the DNA of the paper mulberry, the bark cloth, tapa cloth. And we see by looking at the genetics of the tapa cloth, that does trace back to Taiwan. And we see it being brought into near Oceania, and we see just kind of interaction spheres there. But then we see this lineage out here in remote Oceania, in the Hawaii zone, and lots of interactions and trade and movement of the paper mulberry plant. So this just came out last month, and we've got another paper we're just about to submit where we've been able to get DNA from the tapa itself. The paper mulberry plant is from plants that are living today, and we see both the reintroduction of Chinese varieties coming into Hawaii with the Chinese migration workers that were coming out to Hawaii, but now we're looking also at the DNA, the ancient DNA of the tapa cloth itself, and investigating the genetic variation there. Based on this evidence of connections with South America, I got funding for a major project to go back to Chile and try to find Kiore and maybe human remains. If we find evidence of actually Polynesians in Chile, then we could kind of end that discussion and those debates. So working with Chilean and Rapa Nui colleagues, we excavated on a small island called Isla Mocha that's located 100 kilometers south of where we found the chicken bones there. There's Santiago. Chicken bones were found here. Here's Isla Mocha. Here's another island called Chiloe that I'll talk about in a minute, just on the south coast. This is about 30 kilometers off the coast, Isla Mocha. And we did some excavations, and we found the remains of a burial of a man of my size, and next to him, around his neck, he was wearing a tokikura. Now, this is the term that the Mapuche people call this artifact. They call it a tokikura. This was lying, it had swung to the side of him. As I say, he's my size. We say he was calling to us. His knees were sticking out. He was eroding out of a site, and he had knees that looked like Jonah Lomu's knees. They were massive. He was a strong, robust man, and here he had a tokikura, which was not made out of ponamu, but it's made out of a local serpentine from the mainland of Chile. But they call it a tokikura. Other interesting things we started investigating, and this is very much due to my colleague Jose Miguel Ramirez, stone plank canoes, the way that canoes are constructed in the Pacific. The Mapuche in Chiloe and southern Chile also have stone plank canoes. And the dates, the radiocarbon dates on the canoes that have been found in the rivers there date to about 1350 BP, the same time that we have chickens and the same time that we would expect Polynesians would reach South America. So, and it's the kind of canoe technology, the stone plank canoe technology, we only see it in southern Chile or up in the Chumash in northern California. And some linguists have linked the terminology to sew to Polynesian languages, and the languages of the Chumash and the Mapuche. We've also been looking at ancient DNA of dogs. Interestingly, we see the dogs from the Kuri here linked back to Hawaii. We see the same DNA lineages in the Cook Island and the Hawaiian dogs. We can trace those back to mainland Asia, but interestingly, there's no dog bone in any Lapita sites. So the Lapita ancestors who supposedly brought Kuri Kuri appears as soon as Polynesians are moving out into the Polynesian Triangle. There's Kuri bone everywhere. And linguistically, it's linked within Polynesia, but doesn't link back to Proto-Oceanic languages. So there's some interesting things that are starting to tell us some interesting relationships. Human DNA links Polynesia and Micronesia at the whole genome level. Micronesian origins, we've heard about this today. Te Rangihiroa talked about Micronesian connections. But we've always had Lapita, the Lapita connection, the link. And we see other genetic markers that do tie us to Lapita populations in Polynesia. But our East Polynesians really derive solely from West Polynesia, the story that we've always been told. The animal studies, the Kiore and the chicken, show two lineages, two arrivals of different lineages of Kiore and chickens. And often, both with the Kiore and with the chicken, we see links between East Polynesia and Micronesia. No dogs. We lose pottery. There's no pottery in the atolls. And we see other changes in the material culture, the adzes and the fish hooks, with that expansion into East Polynesia. And this led a colleague and I, David Addison and I, to propose an idea. And it really struck us when we saw this map. So there was one final sea level drop about 2,000 years ago. So this is what the map of Micronesia looked like 3,000 years ago. You have the high islands of Palau and Yap, and the high islands of the Marianas, Chuuk, Ponape and Koshrai. But the final sea level drop at 2000 BP exposed all of the atolls. There's now no people living on these atolls. They're open for people to settle. And it basically gives you a stepping stone through the Pacific and directly into East Polynesia. And we suggested perhaps there was another movement of people that inspired this final push out into East Polynesia, possibly or at least visibly through the atolls of Micronesia. I'm not saying they didn't go to other places, but they're coming from a similar location, a similar further back homeland. So we've been testing this idea, and some interesting DNA results are coming out. This is from Vanuatu, from Teuma, the earliest Lapita site in remote Oceania that was excavated between 2004 and 2009. They found the burial grounds of more than 70 individuals. None of them had their skulls in place. The skulls had all been removed after they had decomposed. We found their teeth where the skulls were, but no skulls. However, there were 5 skulls in the site, all in ceremonial contexts, one of them inside a Lapita pot with another Lapita platter on top of it. Some researchers in the US and in Germany were able to get DNA from those remains. This is the DNA of the Lapita skulls, 3 skulls from Teuma and 1 from Tonga, from the site of Talisiu. This is what we would expect in terms of the possible connections, Lapita connections to near Oceania, to New Ireland and New Britain, and to remote Oceania. The Lapita ancestors are here, next to the East Asians. How do we explain this? They look like they're Asian populations in these first Lapita people, not the mixture that we've always expected based on the material culture and the language and what we know. This was in 2016. In 2018, 2 papers were published that looked at a whole bunch of ancient DNA that was just a few hundred years after the date of the Teuma site in Vanuatu. And what they saw was the Teuma crania looked completely like they're coming out of island Southeast Asia. No mixing with people in the Bismarck Archipelago, the Lapita populations there. But within 200 to 300 years, after the Lapita arrival in Vanuatu, the Ni Vanuatu people look exactly like Ni Vanuatu people today, that have very, very strong, mostly New Guinea or near Oceanic DNA. So they argued we had a complete replacement of the population in Vanuatu. I haven't bought this, and I think that there are other explanations. Are the Lapita skulls really representative of the people in the cemetery? We know that there's ancestor worship and skull shrines and things. You know, is that all the variation that is there? Are these the ancestors? Are they other people that they're putting in as offerings? I think we have to be very careful before we reject this. But I think this interesting idea that we have not been looking at Micronesia enough, both from the Lapita period and from 2,000, 1,500 years ago, to really understand the complex nature of people moving around in the Pacific and interacting. The language of love is universal. People, if you're in a place with other people, you're going to exchange genes. I can't buy that we have, these populations living for a few hundred years in an area without exchanging genes while they're there. So I think we have to be very careful to take all of the information and weave it together. And finally, I just want to say, when we talk about our origins here, after all of this work that we've been using Commensals, I was very humbled to be invited to be part of a project working with Rangatane o Wairau on the Wairau Bar koiwi, the tupuna. And as Rangatane requested the return of the tupuna to be reburied and repatriated at Te Pokohiwi Akiwa, we have a collaboration with my colleague Richard, who spoke earlier, and Rangatane, that they asked for archaeological excavation to identify a place to rebury the tupuna, and they asked for a full biological analysis of the tupuna before reburial, including ancient DNA analysis. And so they were brought down to our lab at Otago, and we've been studying them ever since in collaboration and cooperation with Rangatane o Wairau. And we have found the mitochondrial lineages there. We found much more diversity, all related, all ultimately B lineages, maternal lineages tracing back to island Southeast Asia and mainland Asia, but significant variation when we started looking at the fine grain analysis of the DNA. This is the face of Auntie, burial 1 from Wairau Bar, reconstruction of what this woman would have looked like. And we see these different mitochondria, different maternal lineages, which means these were not sisters or aunties. They were whānau, they were family, they're Polynesian, they're Hawaii, but more variation than we expected. We also started studying modern mitochondrial DNA variation, and we've identified now 30 different maternal lineages, all of the same bee family, the same branch of the tree. All of these lineages are also found in the Hawaii populations. We see these same lineages in Raiatea, in Huahine, and Rarotonga. So there is no Maori-specific mitochondrial DNA or iwi-specific mitochondrial DNA. They're lineages that are shared between the people of East Polynesia, and some of those going all the way back to mainland Southeast Asia. But what this tells us, these 30 maternal lineages, is this evidence of a mass migration. These are people coming from multiple islands within the Hawaii zone. This view of the starving people landing on the shores. These people had their waka, planned voyages with all of their important things and people with them when they arrived. These were not haphazard people who got blown off course and landed here. One of the interesting things we're finding now is a split between East Polynesian and West Polynesian. This is whole genome data collected here in Aotearoa from Pacific, and a large study that we were doing about all New Zealanders. This separation between East Polynesia and West Polynesia is a very interesting thing. And when we now put that East Polynesian data into that Lapita data that I showed you earlier, we see here the East Polynesians. This spread down here is basically European admixture bringing this. So this is where ancient pre-European Maori DNA sits. These are the Lapita skulls here. This is West Polynesia and the Polynesian outliers, and there's a very interesting linguistic story which I'm happy to talk about at some other time. This is Near Oceania and post-2000 BP Vanuatu. Interestingly, this is the Solomon Island Isabel population that's sitting in the middle, and I'd love to figure out, these are modern samples. But one of the important things, and I'll finish here, is what does this mean? It's interesting to address origins, but and this was a question that Rangatane wanted us to address in our DNA studies. How can this help our people today? What DNA evidence can you bring that will actually help us address some of the problems that our people are facing today? And metabolic disease is one of the problems so that's gout, diabetes, cardiovascular disease. And so we can see evidence of that in the archaeological record from Rangatane, from Wairau Bar, from Teuma, from early Micronesian sites. People are suffering from gout. People are suffering from diabetes. And if we could understand the relationships, we actually think there's a reason that we find these high rates. This negative impact is probably because it was advantageous to the ancestors in the past. And now, today, in today's environment, it causes gout and diabetes. But we think it's related to malaria. It helped them survive malarial infections by being hyperuricemic. And so we're testing this right now with colleagues from Guam. So we're working with people in Micronesia, with colleagues at the University of PNG, and here in Aotearoa with a large data set that we have for Polynesian populations to try to understand the evolution of genomes and understand why. And in understanding that, we can perhaps get people to engage. It's not your fault. It's not whakama, you'd be behaving badly, eating the wrong foods and not exercising. But this was something that was important for the survival of your tupuna, of your ancestors. And now we can deal with it with medication and proper treatment, but it's understanding. So, kia ora mai.
Tēnā koe, Lisa, Professor. He pātai, we've got room for a couple of questions.
Lisa, sorry, I noticed you've got the Te Hui bottle gourd on one of your maps, and because it occurs both in South America and in Southeast Asia. Has there been a... Sorry, kia ora.
Yep.
My apologies. I was referring to one of the images Lisa had, which was about the
Hui.
bottle gourd, Te Hui. We know that it's got a lineage in Southeast Asia, and we know there's a lineage in South America. It's one of the most widely dispersed plants in the world. And I was just wondering, has there been any decision about what got here?
Yeah, kia ora. Yes, young researcher, Andrew Clark, has been doing the DNA of the hui, and it is the South American variety as well that came out here. And why did they not need pots? Perhaps they had gourds, they had other ways of carrying and cooking, carrying water and things. But the hui, like the kumara, is coming out of South America.
Lisa, it's been so lovely to see so many of the tipuna of our efforts on both the Māori side and the scholarly university side being acknowledged in these presentations. And it was so great to hear Te Rangihiro's name being mentioned, but I'm also thinking about Ta Apirana Ngata.
Aye.
And for about 30 years, those two, they kind of Apirana ran anthropology in New Zealand, funded it. And they had this dream, I think, between the two of them, of taking the wānanga, the ancient wānanga, and we've had some of the tohunga from the ancient wānanga here, and combining that with the contemporary wānanga and using the research of the ancestors who came on these great journeys right across the Pacific and those who came later, and bringing those strands of inquiry together. And when you think of Te Rangihiro, for example, the way he said, Aotearoa is an archipelago of islands linked by land and not water. And the way the journeys carried on after the arrival of the first ancestors here, when the rivers weren't clogged up in the way they are now with sediment. You know, so many river journeys as well as journeys on land. And Api, with his idea of, he wanted to rewrite the history of our country through whakapapa. Mm-hmm. And he had this dream, and I'm just thinking, listening to you, how fascinated he would be to hear DNA and whakapapa coming together. So it's a comment, but also wanting to pay homage to those tohunga of the past, because I think they are the real inspiration in many ways for the scholarship we're all doing now on voyaging.
Indeed, thank you. Yeah, I'm not saying anything that people didn't know through whakapapa, but it's interesting that now the stories we're telling the same stories, and you don't need the DNA to prove this, but they're telling us these stories, and sometimes they're telling us, and you go, oh, that's interesting, and then you'll find the explanation for what you see. Thank you.
We have Mori and Miriam here. I wonder if they have a quick comment and I'll come
Just a
back to
quick
you.
comment. It seems to me that Roger Green has had an immense importance in New Zealand archaeology.
Yes, yes. Yeah.
Earliest DNA in Aotearoa.
Hi, Rob.
Earliest DNA in Aotearoa you collected.
We have collected DNA from Wairoa Bar, which is one of the earliest sites. We're not collecting DNA as much as being gifted DNA. So if people want us to study the DNA of their tupuna, well, then we will engage with that. We'll try to help answer questions that they might be interested in asking. So I'm not collecting DNA. So I think there are sites, as we've heard, all over Aotearoa that date to this period in time, and I would be willing to guess that they all have the same DNA lineages that we're seeing both in Māori today. We have continuity, continuous, continuity from the first people who arrived here all around Aotearoa to people living here today. So yeah, I guess the oldest that we have is from Wairoa Bar at this point because we were asked by Rangatane to do that, and so I hope that answers your question.
Just to carry on with what Ian was talking about with Raimana Mata. So your kōrero about the kumara and the kuri, those all I kind of reiterated, or the first time I heard about it was looking at the Motete series and learning Motete in there, Pōpō, whakapapa the kumara and tukukuri. So yeah, it's really cool to see the DNA proving all those kōrero, because I really believe that Motete are the way that we retain our knowledge. So,
Kia ora. Kia ora.
Kia ora. We were just curious, we've been doing some studies up in around the Marawana area, and Waitaha is an area that is of huge significance to us, and would be really interested because a lot of our whānau that whakapapa to Waiariki hapu that were established there, and so it'd be great to tee up time to see some of the DNA connections, some of the oral history recorded in some of the Te Raroa archives is around the arrival of the waka Waitaha there, and some of the archaeological evidence which states that the arrival of the Waiariki whānau preceded Kupe. Now there we go, there's one for the books. But preceded Kupe's arrival a few thousand years. So we would love to actually take up time to actually try and get some people to come and check that out for us.
Kia ora.
Yeah, leaving the Bay of Bengal to people and all the rest of it. So kia ora.
Kia ora.
Kia ora, last one.
Kia ora, Lisa. Seeing as it's come up a couple of times today, I want to just sort of reflect on the term mass migration, particularly in relation to the fact that we're not that far away from having got rid of the idea of the Great Fleet. Certainly, some people have picked up on the idea already of mass migration, or the comments around mass migration as echoing ideas of the Great Fleet and that. And I suppose, when I look at it, if we're looking at mass migration, I think Richard was saying sort of like, people moved out here rapidly in a short span of 100 years. If we look at sort of 1,000 people coming out over the course of 100 years, it's a waka of 20 odd people arriving every second year. That would probably equate to kind of like the sealers and the whalers early on in the occupation of New Zealand. And so I'm just curious around this, I wouldn't call that mass migration. And I'm just curious about, sort of the use of the term, or sort of if you'd like to clarify sort of ideas around the use of that term and what you actually imply by that. Sure.
Kia ora. So when we talk about the mitochondrial DNA evidence, so they have done calculations, mathematical calculations, and the probabilities of, given the variation seen, 4 lineages, they've been able to reconstruct back through time. That would have meant, initially, they were saying 100 women in the founding population. That was with 4 lineages. Then with 8 lineages, they were saying, it looks like, several hundred. We've now identified 30 lineages, mitochondrial lineages. And so I think, I'm going to sit down with mathematicians. We're going to start crunching all of the numbers now and modeling this with more sophisticated tools and modeling. And now we're getting whole genome data. I think we're talking about, perhaps 1,000 women. If there are 1,000 women, we're talking 2,000 or more people coming. The DNA can only tell part of that story. What goes with that mass migration picture is based on the archaeological sites around the country. The number, that you see them, all the way around New Zealand, with some very large central ceremonial sites located in strategic locations around the islands. So, it is, how quickly did the population grow? We have the estimates, and lots of debates about the estimates of the population size when Europeans arrived. How mass is mass? It was a large migration. I think, multiple sources, multiple canoes, people interact, arriving and exploring and settling the island. I think maybe Richard can also add to the archaeological evidence.
Just a little bit.
Yeah.
Two things. We've been a little bit conservative with our interpretation of the radiocarbon dates. It's my view that there are actually almost certainly not masses of people here before about 1420. And I think there's independent evidence from the islands to suggest that people have stopped living by about 1400. So I would say we're probably talking about 60 to 80 years, although because of the uncertainty of the radiocarbon dating, we tend to smear it out a bit more. But perhaps a couple of thousand people, as Lisa said, or less, within 60 or 80 years. But one of the reasons why we use the term mass is, first of all, we wanted to get the word migration back in there. Because agency, people were talking about the role of environment, the role of changing wave patterns, and all of this stuff. And we're forgetting about human agency. So migration is about decisions people make. So I wanted to have the word migration in there. And mass migration is just to emphasise that it's a social movement, right? It's not a decision from one tribe or one small group of people. This is an idea, a concept that is going through Hawaii all the time. It is a mass movement in the ideological or ideation sense. Maybe it's not mass migration in that clear physical sense that you're referring to, but I really like the idea of the agency, ideas, mass. I think that it's driven by concepts and ideas that are flowing through this really dynamic social zone. It's a mass movement.
And not outcasts or, yeah.
But isn't this where the wānanga comes in again? Because, there are so many, the Great Fleet's only 7 or 8. But when you go into the oral histories of different parts of, certainly in Northland, but other parts of the country, there's heaps of waka being mentioned. It would be really interesting to kind of figure out of all those waka that are mentioned in the kōrero, the ancestral kōrero, whether it fits in with 2,000 individuals roughly. I mean, I'm just thinking, because that was the dream, I think, that Api and Tarangihiroa had, was to be able to take all these different lines of evidence and weave them. And amazing how they do over time. They start
Come together,
joining
yeah,
up.
yeah.
Last question here with Monica. It's good that we have this one as the last question because we're not running short of time. Our next speaker is unable to be with us. So I'm going to make a quick one with this one and then we'll call up the speaker after that. Kia ora.
Kia ora, Bob. I'm kia ora to the speakers. Look, I was just going to share the same view. I wonder if some of the terminology of the past is still confusing us, like the idea of mass migration. I found from my own research, I've had to get rid of mass. When I start thinking of waka by waka, and stop trying to put dates and times in there, and acknowledge the phenomenon of people moving from East Polynesia moving south, then the words that follow your analysis changes. And it's not mass, it's just watching a migration flow done over a long period of time. It's still happening, actually.
It is still happening.
The Pacific is still moving,
Yeah.
many to Aotearoa, to Australia. So what's new? And so why don't we just study the human phenomenon of people moving around the Pacific and setting up communities?
Which is part of the human phenomenon of people moving across the globe. And I think we can look at that. Why do people go? People always move for new opportunities. And
One of the problems is I wonder whether we've been using European models of migration, and we're trying these short distances. Here you're talking about people moving off 3,000 kilometres or 5,000 kilometres. And we forget the scale of things. And that's quite different to thousands walking from Hungary to Poland.
Exactly.
That sort of thing. So it's, I wonder whether scholars, all of us, have to start reusing more appropriate terms.
I think you're right.
We had a speaker Commensal out and we've actually used up his time. And we're just going to go on to the one after that. So I'll give you one more question, and then we've got to move on to the next speaker, please.
I apologize. It's such a great story. Kia ora.
I'm just saying with apartheid, and I don't know, it might be far out, but in our ancient history and waiata, we whakapapa to the whenua, whakapapa to te wehiwehi, i ka tere, in our, this is our traditional history, so I just wondered if you've ever thought to maybe see if there's any connection between human DNA and reptilian DNA, birds, bugs, I don't know. No?
The great tree of life. We can, DNA unites us all living things, down to the insects, down to, everything. And that the DNA tells that story as well as whakapapa. It is, yeah.
Tēnā, Lisa. It's been fantastic, obviously, with all the questions. And probably if I could just make a couple of comments. I did get malaria when I was in Malaya. And the other part of it is that I'm working in the repatriation of human remains at the Auckland, I should say University, Auckland Museum, and that is my role. And so, I've been told that DNA analysis is quite destructive. And so, we're trying to find out how we get this tupuna is matched to a certain place, a certain area. It's quite a destructive process. Can you comment, please?
It is a destructive process, and that's something we have to acknowledge. People have to make informed decisions about what they want. We can work with a tooth. We can even, a single tooth we can get DNA from. We can get the best DNA is either from teeth or from the inner ear bones. but it is destructive. We can combine the DNA analysis, and DNA breaks down with time. So some samples we might get good DNA from, and other samples we don't, and we won't know until we actually do that destructive analysis. So, but we can combine it with isotope studies. We can combine it with studies of the plaque that your dentist scrapes off your teeth. Well, that calculus that accumulates on the teeth of people in the past, we can study the DNA in that of both the people and the food that they ate, the bugs and things that they carried with them in their mouths, and the bacteria in their mouths that caused disease or ill health. The technology is changing all the time, and we're adding these new tools to our toolkit, and we're hoping, maybe we can start to develop some non-destructive methods. But I never thought we'd be doing what we can do now when I started the study. Things are changing so quickly, and we have to continue these conversations and make these decisions together. So.
Appreciate that, and on behalf of us all, we want to say thank you for your presentation. It's certainly been enlightening to a whole lot of people, and especially to myself. So, ngā mihi kia koe, tēnā koe, mō kōrero katoa kia tātou. Ho mai te paki mo ringa.